Hello everyone, we are solving question number 28 of GATE 2012 and it says match the entries in group 1 with those in group 2. So we have four entries in group 1 and we have to match them with their respective enzyme in group 2. So let's solve them one by one. So first one is MTT. In MTT, so MTT is a salt. It is a 3,4,5 dimethyl triazole 2-vinyl 2,5 diphenyl tetrazoline bromide. It is a tetrazolium salt that is commonly used to detect reductive metabolism in cell for viability, proliferation, and cytotoxicity. I say so. What MTT does, it actually tell directly the viability of cells. So let me just use the pointer. Okay, so what does MTT do? MTT, when react with the active cells, there is an enzyme that is present in the mitochondria, that is succinate dehydrogenase. So succinate dehydrogenase is the enzyme with which MTT react. MTT is reduced in the mitochondria of the metabolically active cells by succinate dehydrogenase, and it yield to water soluble formazole. So it color the color of MTT, that is yellow, is converted into purple. So that's how we get to know the whether the cells are viable in the sample or not. So for MTT, it is succinate dehydrogenase. Moving ahead with the next one, that is annexin 5. It is a cellular protein in the annexin group. In flow cytometry, annexin 5 is commonly used to detect apoptotic cell by its ability to bind to phosphatidylserine. So we have our cells um, outer layer is made up of it is a bilipid layer that where we have phosphatidylserine. So annexin 5 bind to it, and it is a marker of apoptosis when it is in the outer leaflet of the plasma membrane. So what happens when your cell undergo apoptosis? The cell membrane loses its integrity, and all the phosphatidylserine come to the outer membrane, and annexin 5 bind to it, and that's how we get to know how many number of apoptotic cells are present in your sample. So for annexin 5, it is phosphatidylserine. Next, we have methotrexate. It is an anti-metabolite. It is most commonly used in chemotherapy and as an immunosuppressant in autoimmune disease. It treats cancer by slowing down the growth of cancer. It also treats cirrhosis by slowing down the growth of skin cell to stop scales forming forms of the scalps that are formed in cirrhosis, and it may be used to treat erythritis by decreasing the activity of immune system. So what's the action of methotrexate? It actually uh, inhibit the activity of dehydrofolate reductase. So folic acid cannot be converted into tetrahydrofolic acid. So it is the inhibitor of dihydrofolate reductase by folate antagonist. It actually compete with the substrate on the enzyme and result in the deficiency of cellular poles of thymidine thymidylate and purines, and thus in a decrease thus it is involved in decrease in nucleic acid synthesis. So therefore methotrexate interfere with the DNA synthesis, repair, and cellular replication. So when we talk about methotrexate, it is basically the enzyme dihydrofolate reductase, to which it is a direct inhibitor of this particular enzyme. So for methotrexate, we have dihydro uh, folate reductase. Okay, next one is Taxol. Taxol is actually another name of paclitaxel. It is the most well-known natural source cancer drug in the United States. It is derived from the bark of Pacific yew tree, that is Taxus berrivifolia, and is used in the treatment of breast, lung, and ovarian cancer, and is also uh, used in many sarcomas. So what it does, it actually affect the depolarization of it inhibit the depolarization of our microtubules. So Taxol is directly related to microtubules. So Taxol inhibit the depolarization, and that's how there is a um, uh, cell division arrest, so mitotic arrest, you can say. So that is what it does. So So let's match it here. So for MTT, it is succinate dehydrogenase. So P2 we have in B and C. Then we have annexin 5. Annexin 5 is phosphatidyl serine, that is Q4. So Q4 is only in B. So you already have your answer, but let's see other options. Then we have methotrexate, that is dihydrofolate reductase, that is R1. And then we have Taxol, that is for microtubules, then S3. So answer here is option B.